welcome everybody from wherever you are in the world. As Eli was saying, this is the WPC has always been trying to bring people together. We've been doing that since 2006, and we're very keen to get everyone involved. And I'm sure on this research spotlight, we'll have people from very different backgrounds. So we're very grateful for you joining us. As we say, this is the last one of this particular year. And as you know, what we like to do is we like to take people who are working in the Parkinson space and explore a little bit about their life, how they got into Parkinson's, what they're working on. And we're always very keen to have questions. And as Eli says, do put those questions in the chat, and I will try and ask them as they appear at the relevant time. So I'm delighted today we have two, yes, not one, but we have two. So we have the wonderful Tansey, who has been working on Pugsies for many years, very active in the WPC, a vice president of the WPC. So it's the vice president talking to the president, but this is very different from your politics in America, I hope. And we've also got Janna Jernigan, who's joined us today, who is also recently become a doctor. So I don't want to put us down, Mally, but we're not necessarily just starting our careers. So we're a little more senior. So we've got a mix here of people who are a bit more established in their careers versus people who are a little more junior. So I think what would be great to begin with is if I could start with you, Mally. So I'm very interested to know your origin. So where did you begin? And then I'm interested to know how you got into working in Pogsy. So
yes, of course. Thank you for having us. It's a pleasure. And thank you everybody for joining. I would say that I was interested in science from, grade school, really curious, had a wonderful biology teacher in El Paso, Texas, Margaret Jackson, who's still around and is amazing. And she encouraged me to go to science fairs. I went to an NSF, National Science Foundation, summer program at UC Davis, and I learned a lot about the genes that make wheat resistant to drought. That was a long time ago, and so I got really interested.
So this was when you were at high school?
Yeah, it's a high school level.
Okay. Yeah, great.
So then I got really interested, and I decided to pursue a degree in biology. I went to Stanford, got my master's and my bachelor's in biology. And then from there, I was one of many pre-meds. I wanted to go to med school.
Nothing wrong with that.
Nothing wrong with that. But what turned out is that in the application process, I applied to Texas schools, and there were 6 Texas medical schools at the time. And my soon-to-be husband also was applying. We met at Stanford, and he got into one school, and I got into 5, but there was no overlap. And so I said, well, let's go to Dallas. You start at UT Southwestern. I'll work for a year, and I'll reapply. Because he had already applied in California and not gotten in the year before. So that basically opened my eyes to research, and I was super excited to work as a tech for a year. And I decided, no, I want to do research. I want to, be a PhD.
So when you were doing your degree, your original degree, that was more sort of lectures, library based. You didn't do any summer projects or things in the lab?
No, I really didn't at Stanford. I did in my master's year, I did some research at the Hopkins Marine Station down in Pacific Grove, California. And that was wonderful. So that was like a master's thesis research project. Yeah. And from there, I, got interested in, excitable cells. I worked on smooth muscle contraction, how, the smooth muscle in your body works. And then I got interested in neuroscience. My grandfather had Parkinson's. Okay. And I decided, I want to understand it. He lived in Mexico. He was never officially diagnosed or treated, but he basically went through all the symptomology. And when I look back on it, it was very, very typical, progressive. And I went to postdoc in St. Louis and learned about GDNF and other neurotrophic factors that.
So was this in the year when your husband to be, or may have been a husband, was now at medical school?
That's right. He was doing a residency in St. Louis, and so we went together.
Okay.
He did an MD PhD and I did a PhD, both from UT Southwestern. And then we
Okay.
went to St. Louis to WashU for his residency, and I did a postdoc. And
Okay.
after that, I realized, I really love this, but I want to work less hours and maybe more money. And I decided I would go to biotech. And let
Okay.
me tell you, I learned a lot, but it's not stable, right? And so,
Yeah.
this company gets bought by this company, and there's a lot of turnover. So I felt like I really wanted more stability. And the most important thing is I missed the mentoring of the students. And so I went back to academia after 2 years in biotech. And
And during this time, Manu, you'd already declared yourself as being interested in Parkinson's. Apart from the family connection, the
that's
work,
right.
what
I was
excited you
working.
was the
Yeah,
work around Parkinson's, and that was about growth factors for restoring brain function.
that's right. That's right. I was in the lab where we discovered nurturin,
Oh yeah,
the sister ligand to GDNF, right,
yeah,
which also went into clinical trials by Sarah Jean. Um, and so in biotech, I learned about inflammation. I was
okay,
given this project to work on this next generation anti-inflammatories, and it opened my eyes to maybe there's a role for inflammation in Parkinson's. And
So
that's
this,
how I
so this
started.
work on anti-inflammatories was not in Parkinson's disease. It was just an anti-inflammatory
No,
drug for arthritis or nothing
exactly.
really.
Yeah,
Yeah,
it was being
yeah,
developed for peripheral applications. And
right.
when I left, I said to them, I would like to take these research tools to ask questions in my lab
Yeah,
about whether inflammation plays a role in Parkinson's risk or progression. And that's
okay,
how I started with a grant from the Michael J. Fox Foundation in 2003. So it's
okay.
been 21 years.
Okay, well that's so basically decided for domestic reasons not to go to medical school, which is great for the scientific community because who knows what might have happened. You then went and did your research, sort of defined yourself around Parkinson's, then go into biotech where you earn a lot more money, but miss out on students and I would guess a degree of academic freedom. I mean, I don't work in biotech, never have, but
Definitely.
I imagine it's quite focused on what you need to do.
Yeah,
And then you came back into the lab to set up your own lab. And I think these are very important times because obviously those first grants are the most important because you've got no track record, no disrespect. But I mean, we all start somewhere and you basically
that's
think, I,
right.
I
Yeah.
haven't really got a track record, but you're going to take a bit of a punt on me, a bit of a gamble. And and so you started in the lab looking at inflammation. And how big was your lab at that point? And how big a shock was it to drop that massive salary that you had in biotech for
Yeah,
the
right. I took a pay cut, of half a pay cut. It was difficult. I started out with three people,
yeah,
myself, a technician, and one student. And we actually did start out working on GDNF. And all of a sudden we realized, no, we really need to work on more inflammation. And that's when we applied for the grant. And
yeah.
Fox put out this, call for role of inflammation in Parkinson's. It was almost like dropped in our lap. And
So this was 2003, which was
2003, exactly.
so
Yeah,
it wasn't exactly the hot topic of the day. I mean,
it
now
was not.
it's now obviously very much everywhere. But inflammation, I think Pat McGeer had done something in '87 or something. I think.
Exactly. The late 80s, the Edith and
Yeah,
Patrick McGeer were
yeah,
the grandfathers of it all. I think they found the activated microglial cells
yeah.
in the
But there was a sort
brain
of perception at
postmortem.
that time that inflammation was merely a consequence of having the disease. The disease struck, cells die, a bit like you cut yourself. When you cut yourself, inflammation comes up to clear away the damage. That's all it is. But you thought it's more than that.
Yeah, I saw there was a little bit more than that because there were some studies suggesting that people with a lot of chronic inflammatory conditions were at higher risk for Parkinson's and for Alzheimer's. And then the epidemiology studies, which basically do associations, right, showed that people through the many years, based on medical records, that were had these diseases, but were treated with anti-tumor necrosis factor or non-steroidal anti-inflams, that all of them as a group had like a 50% lower incidence of dementia and Parkinson's. So
Mhm.
to me, that suggested that inflammation in midlife may be a risk factor many decades later, and
Mhm.
perhaps trying to control or target that inflammation in many different ways, exercise, activity, medications as needed, may be a way to reduce risk.
Okay. So it was sort of rather circumstantial evidence, if you like. There'd been these
Correct.
observations that if you look in the brain, there's some inflammation with microglia. And then there were these associations that if you had infections, you were more likely to get it. And if you treated infections or these chronic inflammatory diseases, your incidence dropped. So it implied that in some way there was some sort of link between it. So when you were setting up your lab, there's you and two others or three others.
Mhm.
Um, so what was the first project you did? What was the sort of question you asked yourself? Because obviously taking this sort of circumstantial epidemiological data into a sort of testable hypothesis in the lab isn't
Yeah.
isn't obvious, given, as we've discussed on many of these research spotlights, Pug disease is a unique human disorder, and whatever inflammation we have in humans is probably not the same as in rats and mice. And
Correct. That is what keeps me awake at night, that the immune system of, rodents is not the same as humans. However, using rodent models, which have been used for many years in immunology and cancer and, neurodegenerative disease, we basically ask the question, if you take a mouse model where you can model some of the dopaminergic neuron loss induced by neurotoxins, induced by inflammation, can you then interrupt the inflammation early on or later on? And can you protect those dopamine neurons? And that was just a simple question that we did with the Michael J. Fox grant. And it turns out that you can. If a rodent model is destined to have, say, 80% loss of dopamine, if you interrupt the tumor necrosis factor pathway, you don't get to that threshold of 70 to 80%. It is reduced by 50%. So mechanistically, in a controlled experiment in the lab, if you block the tumor necrosis pathway, which induces cell death, we've known that for quite some time, and promotes inflammation, you can basically arrest the progression. So that's in a mouse model. And so
okay.
the real
So the
test will
argument would
come.
be it's not necessarily true. So Pog disease happens because it happens. I
Yeah,
mean, it may be inflammation that starts it, but the
yeah,
argument from that data would be whatever causes Parkinson's to begin, there is clearly an inflammatory component, and that
correct.
is driving the disease.
That's
And
right.
this tumor necrosis factor, I mean, where does that come from? Does that come from the circulation diffusing into the brain, or is it produced by cells in the brain, or is it a combination
Interesting
of the two?
question. Yeah, it's very important. It is made by inflammatory immune cells in the blood, but it's also expressed or on the surface of neurons. And when
Okay.
neurons are injured, they release it, right?
Okay.
And so it's kind of a signal to immune cells in the brain that there's been an injury and they come to the rescue, right? So the TNF can come from injured neurons. It can come from immune cells. And it's a way to communicate between the neurons and then the caretakers of the neurons, which are basically your immune cells, your glial cells.
So in Pog disease, the hypothesis would be the cells are dying off. They release TNF alpha. There's also that causes some inflammatory response. TNF alpha comes in on peripheral immune cells, and then that drives the disease process. And if you therefore block TNF alpha, in theory, you should be able to arrest or slow down the disease progression.
That's right. That's exactly right. And in fact, it's ultimately a hypothesis that we need to test in the clinic. And
Yeah.
there are some plans on the way for that with various different anti-TNF modalities. And I think that's where we'll be able to see if it works and affords clinical benefit.
Okay, I'm going to come back to this about the
Yeah.
different types of inflammation, but I just want to just before we move on to that, you obviously missed mentoring people. That's one of the reasons you left biotech. So what is it that you like about the lab, apart from the science? What is it you like about that, and how do you bring people on a mentor? What do you think makes a good mentor, or what makes a bad mentor?
Yeah, well, I think the most important thing for me is to have an opportunity to train the next generation of scientists, right?
Yeah.
You mentioned that you and me are, more senior. I like to say more seasoned. And it's our responsibility to train the next generation, right? I want our brains to be in good hands.
Yeah.
And so I think that the joy that I derive from being a mentor is to see, the lighting of the fire of ideas of, hey, maybe we should think about this disease in a different light and come up with different therapies, right? Because how you think of a disease helps you come up with solutions. And
Yeah.
so we think that immune dysfunction in throughout your life may be a risk factor, and that may be something we could fix way before you get into your 50s and 60s, right? And so when I have people like Janna come to my lab and they're curious about how does the inflammation outside the brain affect the brain, that's a new concept, right? That people have started to accept because there's a lot of support for it. And so my mentorship hat has to do with uh, supporting them to develop their ideas, helping them focus it, and then getting out of the way to be able to have them explore their own hypotheses and see if they're going to like it.
Yeah, because I mean it's a wonderful thing to do. I mean I do a lot with junior scientists, a lot with junior clinicians. As I get older, the junior clinicians become much more important because they'll be treating me when I come into this hospital and in the future. So it's good that they know what they're doing. But it is this tension between encouraging and shaping, giving independence, but not so much independence that it's a project that's so removed from what you're actually doing. So, and just before I move to Janna, I want to ask you, Melanie, who are the big people? You obviously had this high school teacher who did a lot for you by putting you in touch with science to begin with, and your mentors, who have been the people that are sort of that you would say have been a great role model for you.
Yeah. The first and most important ones have been my parents. My
Right.
mother was a languages and linguistics teacher at UT El Paso, and she valued education from a long time ago. My father is a certified public accountant, and he, both of them grew up in Mexico. They became residents. My father, in particular, valued education and making sure that his daughters would be independent and successful professionals. So they were probably the most important. And my father, who is 91, continues to be a great mentor, and he's helping me edit a book about my trajectory in life as a Latina in STEM. Um, and he's written like 6 books on the family business, which is what he was involved in when he was before retiring. And the second mentor that I really end up talking a lot about, maybe Janna can tell you, is Gene Johnson. Gene Johnson, my postdoc mentor at WashU, who was involved in the discovery of nurturing Persephone, Artemin, all the GDNF family. He's still around, and we're actually getting together as a lab in April. He taught me a lot, like, don't look over your shoulder, don't worry about the competition, do the best experiment you can, share your ideas, don't be so protective, because if you're protective of your ideas, people are going to think you have no ideas, and that's worse than
Yeah.
someone stealing your ideas, right? Ideas are cheap, it's the execution that matters. So I think that those are the people I would put on there. And my graduate mentor, Jim Stoll at UT Southwestern, um, he put up with a lot of abuse, I think, from 3 female graduate students. We were all, pretty vocal. But he definitely was one of my formative mentors.
Yeah, well, that's great. I mean, Miguel Vela has been with us, visiting us today, and he did the very first research spotlight. So I was talking to him about it and saying that, to me, clinically, David Marsden, the late David Marsden, and Andrew Lees were neurologists who gave you a great deal of time and support in a point in your career where you were merely a medical student. So why would they give you why would they spend any time with you? But actually, being generous with everybody is a very good, doctrine for life, and it was very helpful for me. So, Janna, where did you begin life, and how did you end up on a research spotlight on the 3rd of December
Yeah, well, so I grew up in Georgia, and
in
just
America?
yes, America.
Yeah, just
And
yeah.
yeah, and I just like Malou was always very science minded as a kid. I knew going out of high school that I wanted to jump into an area that was kind of still a mystery to us, like the brain or the deep ocean or maybe space. I don't know. I was all over the place at that point in time. But I ended up getting a concussion my junior year. And it was decent enough that I had trouble reading for a little while. Thankfully, though, I recovered. I was young. I'm all good. I've managed to get a PhD.
Right.
Um, but at the same time, my grandfather was going through the last couple years of his Parkinson's journey. And what I noticed is that when I had my concussion, I could feel some of my faculties just trapped. And
Yeah.
they were recovering, but for him, that really wasn't the case so much. So I felt like it gave me a little bit of a glimpse into a tiny bit of what he was experiencing. And that really kind of just propelled me into learning more about the brain and understanding Parkinson's. So when I went to undergraduate, I went to the University of Georgia. Um, as soon as I got there, after I finished my freshman year, I jumped into a research lab. And the one that was there was actually the only one working on Parkinson's disease. Happened to be my mentor was Jamise Lee, who was a former postdoc of Malou.
Small world.
So small world, always a
Yeah.
small world. And so while I was there, I got introduced into the idea of, the immune system in Parkinson's and
So what was
inflammation.
your specific project? That
Yeah, so my specific project, I was working on microglia at the time, but we were working with microglia that had lost what we consider a protective factor in the immune system. It's called RGS10. Most people don't know what it is. But I've been
R,
working
say it
on it
again, RGS10.
yes, RGS10.
And this is something that's expressed on microglia, and that enables them to perform a function of clearing alpha-synuclein or an immune response, or
So it's intracellular, so it's inside the cell,
yeah.
and it's really important for regulating the inflammatory response.
Okay.
So when it's gone, you have a higher inflammatory response in these cells.
Okay.
So we were
So your
working.
initial project was you took these cells, it was absent, and then what
Mhm.
did you do with them?
So we put them in different conditions.
Yeah.
So we put them specifically in hyperglycemic conditions. So we put a lot of extra glucose in the solution where the cells were, and we also put a little bit of an inflammatory stimuli in there to sort of understand how when you have metabolic dysregulation, which is
Yeah.
something that's a common comorbidity in Parkinson's disease, how that also might be influencing these cells when that protective factor isn't there. And we found that that protective factor seems to be important for regulating inflammation under a lot of different contexts. So we know that it was important for regulating a normal inflammatory response, but it also seems to be important for helping the cells respond properly when there's metabolic dysregulation as well.
Okay. Okay. So this is this is interesting because I mean, there's been a lot of interest in metabolism and how cells metabolize and how that influences how they behave and that goes wrong in disease. So changes in respiratory pathways and things like that. And
Yeah.
obviously inflammation is whilst it's compartmentalized, is also a systemic response. So then after this project, you then did a PhD with Manu. Is that how you?
Yes, I did. So I started with all of my research kind of like with the immune cells in the brain and the microglia. I did a couple summer research projects also about microglia, slightly different projects. But when I got to Malou's lab for my PhD, I knew I wanted to go there because it was like inflammation central. And I also knew what kind of a mentor she would be because I had a very similar mentor who mentored me in undergrad. So I knew it would all work out pretty well. But at that time, I'd been all up in the brain in terms of inflammation. And when I got into her lab, I started wondering, okay, well, what's happening when you lose this protective factor in the peripheral immune cells?
Okay.
What's happening outside of the brain? And could that be contributing at all to what happens in the brain later on?
So this factor, RGS, whatever it was,
Or
is
just
inside microglia, but it's also, is it in all immune cells in the periphery? So is it in T cells, B cells, monocytes, all cells? So is it universally expressed in all inflammatory cells, or is it universally expressed in all cells?
It is most highly expressed in immune cells. It is
Yeah.
in other cells, but not so much. It's mostly in innate immune cells
Okay.
or myeloid cells, so your macrophages and monocytes. And it's also very highly expressed in like CD4 T cells specifically. And when we look in Parkinson's disease, we look at the monocytes, we look at the CD4 T cells, there's less RGS10 in those immune cells in Parkinson's and individuals with Parkinson's compared to healthy controls.
Okay, so let's now take apart the immune system in Parkinson's. So we've got, you're interested in this factor in immune cells, which is a sort of break on their immune response. And Mally's got this TNF alpha hypothesis. So I'm going to go back to Mally now and just say, how has your work evolved over the years? So obviously you're now getting to the point where you say, ultimately to prove anything, you have to do a clinical, well, I'm not saying you have to, but I mean, if you're going to intervene with the immune system, given their differences, you'd have to intervene and show that if I block TNF alpha in people with Parkinson's or upregulate or whatever you do, but you've got to modify that to show that there has an effect on clinical progression or such like. So in your case, Manu, how has the work evolved over the last 20 years since you did those original experiments? What's been the sort of major shift in your sort of scientific direction in how you have understood CNS inflammation and how you've tackled it in the lab? And then I'm going to come back to Janna to how she's done it in the periphery, and then we'll link the two together.
Yeah, I think that's a really important question. We were very focused initially on the brain. I think the whole field has been very focused on the brain, but now with a lot of different studies and different labs, it's clear that the brain is not immune privileged, right? It sees and senses and is impacted by peripheral organs. It talks to peripheral organs through substances that circulate across your blood-brain barrier and, across your gut barrier as well. The microbes in your intestine are really important to program and direct the microglial cells that Janna was talking about.
Because there was this idea that the brain, coming back to this idea of immune privilege, the brain sits within this cavity. It has this blood-brain barrier, so the blood constituents can't readily get into the brain for very good reasons. And so there was this sort of idea that the peripheral immune system cannot access the central nervous system, and the central nervous system doesn't really access the periphery. So they were isolated. But as you're saying, that's not an absolute immune privilege, it's a relative. So it's relative compared to other organs in the body, but still the immune systems talk to each other. So carry on. You're saying, so there's microbiomes in your gut.
So there's
gut inflammation.
Yeah, every barrier site with the environment has a microbiome. So there's a skin microbiome, there's an oral microbiome, there's a gut microbiome, and that barrier protects you from things from the outside, pathogens and other things that you don't want inside your body. But those barriers, studies have shown, break down over time and with age.
Yeah.
And our focus has been, as you're aging, since aging is the biggest risk factor for Parkinson's, right? There is genetic predisposition. There's also environmental exposures. And I call it the dance card of life, right? You're born with certain genetics. Some of those genetics may make your microglia, which are your professional vacuum cleaners, a little lazy, right? Or some genetics may make them overperform, and you do fine for the first 4 decades of life. But then things start catching up, and your brain and some of your organs sort of become like a hoarder's episode. The trash hasn't been taken out, the vacuum cleaners aren't working, there are fewer of them. And that, we believe, contributes to this toxic accumulation of not just synuclein, but other things that make life difficult for neurons and circuits. So the idea then is to figure out, how can you identify those people that have the genetic predisposition, the environmental exposures, and detect their higher risk before they become even in a prodromal, REM behavior disorder or anosmia. We believe that by looking at the peripheral immune system, because there's this crosstalk that Janna can explain, we can use those as a window into the brain and what's going on with your brain, and be able to use your peripheral blood immune cells and challenge them in the dish to see, are they underperforming? Are they overperforming? One of the recent works from our lab, from Rebecca Wallings, is that when your immune cells overperform as you're young, they become immune exhausted. which is the concept that we haven't really talked much about in neurodegeneration, but it's very well known in cancer. And so there are a lot of analogies, and we believe that immune exhaustion may be one player in the aging process in your immune system that actually contributes to the caretakers, vacuum cleaners, not doing their job, and then neurons are neglected, right? It's not just about neurons, it's about the sentinels and the caretakers of the system.
So the argument would be that if your immune system's been very active, it gets exhausted. And so when alpha-synuclein, which could be conceived as a pathogen, your immune cells can't really deal with it because they're tired. And so it gains a sort of hold on the brain, and then the disease starts to sort of take hold and
That's
carry
right.
on. And then
That's right.
just so I've got it clear before I come back to you, Janna, is obviously you've talked about microglia resident cells in the brain, and then you've talked about environmental. And so your argument would be that the microglia in the brain misbehave in Parkinson's because they're triggered by something in the periphery, which then gets into the brain and starts it. So there's nothing. So you might have microglia that have a different intrinsic activity, whether they're more active or less active, but that doesn't matter unless something happens in your periphery. And something happens in your periphery because of, well, your genetic makeup, but also environmental things. So you strongly believe that there are things in the environment which trigger Parkinson's, or
Yeah, I think there's plenty of evidence that exposures like pesticides and certain metals, some can come through the gut, but they can also be inhaled, right?
no.
So this idea of your body first Parkinson's that's been promoted, it's a hypothesis by Per Borghammer and colleagues, is that if it starts in your gut, you may end up with body first Parkinson's. But if it starts in your nasal epithelium, you may end up with brain first Parkinson's. And the symptoms may be different. The propagation or progression of the synuclein misfolding and misbehaving may start peripherally and going to the brain, or may start in the brain and may go to the periphery. But yes, we hypothesize that things in your gut, like the microbes in your gut, are making substances that are affecting microglia in your brain, and they're affecting the peripheral immune cells to maybe go into the brain. And we don't know if they're going into the brain to help microglia because they're also talking to each other, right?
Yeah.
Or they say, you guys aren't doing your job, we're coming in, right? Because the vacuum cleaners you have in your peripheral blood, the macrophages, really pack a punch. And when they enter your brain, I call them bulls in a china shop, right? With a lot of punch, and they did not grow up around neurons, right? And most of them, they're protecting your tissues and vacuuming up that mess there. And so that entry of peripheral immune cells may be a little bit, of a dysregulated crosstalk. And that's what we're trying to detect in the earliest prodromal stages, so that's the part that can be modified, right? Your genetics, not so much yet, but the way your immune system may respond to those environmental exposures may be important. Exercise is another thing that changes your gut microbiome, physical activity, diet. And so we believe those are the links that are now being appreciated.
So basically, the disease is triggered in the periphery, either in the gut or in the nose. That peripheral trigger, whatever it is, sets up a sort of local response, which then spreads via the immune system into the brain, and then
Yeah,
and then the
exactly.
disease carries on from there.
Yeah, and some examples may be, we know that synuclein in the dish and the brain, in animal models, misbehaves and increases in amount when there is chronic inflammation.
Yeah.
So if you have gut chronic inflammation from perhaps inflammatory bowel disease or exposures to chemicals or pesticides, that could be potentially sufficient to cause the synuclein that's in your enteroendocrine cells lining the gut to increase in amount. And then they go, we don't need this. And they pass it on to the nerve terminals, the ganglia that they're anatomically connected to. And then those are connected to the vagus, right? So we got to talk about the vagus if we're talking about Parkinson's. But the hypothesis is that the synuclein may go from your gut lining to the glia to the vagus and then up through the Brack highway, if you will, into the brain. And
So basically, they go up from the gut, up the nerves, into the
that's
lower
right. It's a
brainstem.
hypothesis, yes.
Yeah.
But
So one of the
things that support that, just real quick, is that individuals with IBD and Crohn's appear to have a higher predisposition to Parkinson's unless they're being treated with these anti-inflammatories or anti-TNF drugs. Then, Inga Peter did some studies, small studies, but they need to be replicated, showing that if you are treated and you have one of these IBD or Crohn's, then your risk for PD goes down by 75%. The other thing that supports this idea of a highway is some studies that show that when you get a vagotomy to be treated for ulcers, right, before we knew how to treat ulcers better, those individuals with vagotomies appear to have lower incidence of Parkinson's. So, it's circumstantial evidence that supports the idea of the communication.
Okay, so just before I come to you, Janna, one of the questions we've had is about how you model this in the lab. So one of the questions we've had from Charles is that, what's the animal model you use of PD, and TNF alpha antagonists are protective. Have you ever tested these antagonists after a significant loss of the dopamine cells? I mean, it'd be useful just to hear about how you model it, and then have any interventions you've done helped. And then I'm going to come to you, Janna, to explain what you see in your model with respect to these peripheral immune changes.
Yeah, certainly. We've used different models to induce either dopamine depletion, like 6-hydroxydopamine or chronic LPS. And in those models, when you treat the mice with an anti-soluble TNF drug, they are able to, that's able to interrupt the loss of dopamine neurons. And so you give this substance peripherally, it goes into the brain, it probably has some benefits peripherally, but it stops the progression. And you have to give it within 7 days of the lesion. If you wait too long, you'll still bring down inflammation, but you lose the window of time to rescue those neurons before crossing that 50% threshold.
Because they've already died by that point.
They've already died, and the inflammation at that point is too late. And this is why I think the clinical trials have failed, because if you start taking NSAIDs, non-steroidal anti-inflammatories, or even anti-TNF drugs, once you have advanced stages of disease, I think it may slow down a little bit of progression, but by that time, it's a little bit late. So the idea in the trials that are being designed now, Charlie, is to go in with individuals that are anosmic and have rare behavior disorder, so that you can do a trial to see if you can keep them from converting to clinical motor PD.
I mean, I think the problem with clinical trials is often they're done too late, and the problem in the lab is often people give the treatment before they give the lesion, and therefore prevent the lesion, which is what Charles is mentioning here. And that obviously would mean clinically you'd have to treat people before they knew they even had a problem, which is not realistic.
Difficult. Yeah, you would need a super safe drug, which I hope we have, and I was involved in developing that when I was in biotech. It's taken 20 years to get to the clinic, but it is being tested in MCI and Alzheimer's in a phase 2. So we are hopeful that we can get it into clinical trials for Parkinson's.
Yeah, and it may be the whole, people are going to go earlier now. We have this alpha-synuclein seed assay, so we have greater confidence in people at high risk. So Janna, come to your PhD and your work. So what was the model you used, and what were the sort of major findings?
So I used a chronic LPS model. So essentially what we did is we introduced a low level of inflammation peripherally for 6
So
weeks.
LPS is lipopolysaccharide. So this is what bacteria have on their
just on the outside of the bacteria themselves. They shed it all the time, and your immune cells respond to it like crazy. They're like, oh, there's a bacteria. So we just put a low dose of LPS in these animals for 6 weeks when they're in adulthood. And then we asked what was going on with their immune system through the blood, and also in and around the brain specifically. But at this time point, we were specifically asking, what is the immune system doing before degeneration starts to occur?
Okay.
And so what we saw is
So
that,
so when you give LPS to these animals, the normal consequence, apart from the obviously inflammatory ones, is this does lead, because Mally was sort of intimating this, it does lead to dopamine cell loss. So it's a model for producing dopamine cell loss in rodents.
yes, it can. Although with lower doses, it takes much longer for that to occur. With high doses, you can see it within like a week. And if you inject it straight into the brain, those neurons will definitely die.
Okay. So you've given low dose LPS, you've got this chronic, you look at then the changes in the inflammatory response or inflammatory system peripherally and centrally.
Hmm.
And I'm going to guess you probably found changes.
Yes, we did find some changes, but we asked this specifically, like what happens in a normal immune system, and then also what happens when we've lost that protective factor again. So
Yeah.
that protective factor from my undergrad, RGS-10, we were looking at it
RGS-10.
again.
Yeah.
Yeah, so we found that there are a decent amount of dysregulated immune responses in the blood and around the brain with just low dose LPS. Like it was sufficient to start changing things after this time period. But when you also didn't have RGS-10, the amount of dysregulated responses, especially around the brain, became much more pronounced, which we
Okay.
think would be important for the development of neuroinflammation and then neuronal degeneration.
So when you didn't have RGS-10, you got a much worse outcome.
Yes. So when you take this accumulating low
Yeah.
dose inflammation and then you top it on with something else, these like kind of gene by environment interactions
Yeah.
that we think of in the development of the disease, the chances of your immune system responding in I'm going to call it a much more messy fashion
Yeah.
start to increase and probably end up making things much more uncomfortable for your neurons at the end of the day.
So basically, if you increase the amount of inflammation peripherally and centrally, then you're driving the disease process, which comes back to something Manu was saying earlier. And this RGS-10, is that druggable? So one of the questions people will ask is, well, if you're saying that's a bad thing, is there a way in which I can target that? And obviously, the challenge here is your immune system is there for a very good reason. So obviously, if you remove someone's immune system, then in your theory, it would slow down Pogsy's, but you would succumb to something else much more quickly. So is RGS-10 druggable? Is there anything you could do to change that? And secondly, how much would you have to change it in order to have a protective effect without making you more predisposed to getting other infective problems or inflammatory conditions?
Yeah, those are great questions. So for the first one, we've been able to manipulate levels of RGS-10 in the lab, so we can do gene delivery therapies.
Mhm.
Specifically, there was one Michael J. Fox funded study where they did a gene delivery of RGS-10 in the brain with one of those PD models, the 6-hydroxydopamine. And when they reintroduced RGS-10, both the microgliosis, so all the neuroinflammation, and even the neurons were, whereas they normally die, most of them survived and the microglia were much more calm. So we know that reintroducing it can be a good thing for the brain in mouse models.
Yeah.
And so there's the gene delivery system. We don't really know too much about like endogenous things that might be able to increase RGS-10 at this point. It hasn't been studied enough. So we don't know necessarily if like exercise or particular things in your diet can help.
Yeah. Or
But there are
small
also
molecules? Are there small molecules you could use with, systemically given things which could target it? I mean, is that because I'm just thinking if this is such a central part of the inflammatory, coming back to Mally's work in biotech, I would imagine biotech and pharma have probably been in this space because obviously information is useful to understand hypoxes, but it's useful to understand full stop. So I don't know if there's any drugs or molecules that people are using to try and target this, if you're allowed to talk about it.
not that I know of at this point.
Okay.
I think
Yeah.
people are just starting to get interested enough.
Okay.
Yeah.
But also we know that RGS-10 is epigenetically regulated, so the cell will wrap it up. The DNA of RGS-10 will get wrapped up upon an inflammatory stimulus.
Right.
So anytime it's really inflamed, RGS-10 levels go down. So if you use something called an HDAC inhibitor and allow for the DNA to be unwrapped, you can increase RGS-10 levels again. So there are some ways of doing it, but that might be a little bit too global of a change to just change the entire epigenetic landscape.
Let me interject real quick. I think the interesting thing, as Janna pointed out, is that this is a regulator of inflammation, right? It keeps the lid on inflammation everywhere in your body, according to many of our studies and others. But it is down-regulated by chronic inflammation. So if you are slowly becoming inflamed as you age, you'll probably start to lose it, right? It may go up and trying to cap inflammation, but then you probably start to lose it. And one thing that Janna did that she didn't mention as part of her thesis is she looked in the Michael J. Fox PPMI database, and they looked at the levels of RGS-10 in healthy controls or in Parkinson's. And Parkinson's individuals have less RGS-10. We don't know if that's because they were inflamed or because they lost it and then they became more inflamed, right? So either way, it's possible that gene therapy or small molecules that bump up the levels may put a brake on things or may, regulate inflammation and help with progression.
But it's obviously a sweet spot because, one of the problems in COVID showed was that actually, whilst obviously some people died of COVID, it was actually your immune response to COVID that was more of a problem. And,
absolutely,
a lot of people who did very badly had a massive response to it. And that was your immune system driving the problem.
It's a double-edged sword. You cannot immunosuppress individuals. It's very difficult to then fight a normal infection. And I think that's the tightrope that we all have to walk. We have to make sure that we don't have untoward side effects.
Okay. And so one of the questions which is coming up, which is very sort of opposite, I think, is we've been talking about inflammation, we've been talking about Parkinson's. What about sex differences? So, Parkinson's is slightly more common in men than women. A lot of autoimmune diseases are more common in women than men. So when you're doing your experiments and when you're trying to think about this, I know absolutely nothing about this, completely empty file in my head on, the immune system differences between sexes, and presumably also, that will change. It changes with age, which we know, but I don't know whether there's greater changes in men or women. So how much do you factor that in? How much do you think that is an important factor?
Janna, you go first.
Okay, I'll go first. So we think it's really important that every time we're doing a study, we're powered for sex because we know that the immune system of both men and women just respond differently. So even in my models, I separated all of my data by the boys and the girls, and I could see that a lot of the phenotypes I was picking up were a lot more prominent in the boys, which was very interesting. So, see, I think it was one of my colleagues' work, Hannah, I believe it was a study out of Stanford. Blue, can you correct? Is that the study? Am I thinking of the right study? Yeah. Where they found that potentially one of the reasons females have more autoimmune conditions is because of that extra X chromosome and the way it gets wrapped up, and that it can cause kind of these immunogenic peptides to be kind of spewed out into the system. But that likely, or I'll just stop there and say that we know that there are major differences, and that some of those sex differences are starting to be uncovered.
Yeah. So I guess the important point here is there are differences, and you factor that into your modeling. So I think traditionally, a lot of people have just sort of, when I started doing research in the dark ages, we used to use female Sprague-Dawley rats for our transplant experiments. And when asked why, you say, well, they just work. And so there was no question of sex differences. You just used whichever model was there and just accepted it. Whereas now, and certainly when you move things to clinic, you have to look at male and female animals. And so Mala, anything else to add about these sort of sex differences in terms of understanding inflammation or its impact in Parkinson's, or is it?
Absolutely. And I know Rochelle Flanagan is on the call, and she's been very interested in this and came and visited us here in University of Florida this year. So one of the things, there's definitely differences. And one of the things that makes sense in terms of evolution is that males and females had to be wired differently, definitely to withstand the pain of childbirth. And childbirth is not just, you know, a necessity, right, to preserve the human race,
Yeah.
but it's also the control of pain is very much tied into the neuroimmune crosstalk, right? And we know that microglia in female rodents and humans are wired differently to respond to pain in a different way. So we know for sure that's one, you know, wired in difference that happens. We also know that while females may be more, you know, inflammatory to respond to something, and that has been shown in lots of different labs, the males seem to be able to turn off or suppress some of those inflammatory responses when they're young.
Right.
When they're older, they may not be able to suppress as well because their immune system may already be exhausted. So it's a trade-off, right? That if you suppress too much or you become exhausted because you have such a good immune system, then later you pay the price. It's a hypothesis. We're obviously exploring it. And as Rochelle pointed out, there are countries where the incidence of Parkinson's is higher in females than in males. I personally am not that impressed by the sex difference that we all talk about. Oh, slightly more males.
Yeah.
It's really not that much, right? And I think it's more of a gene by environment interaction. The idea is that you want to have lower inflammation all your life. And the best way to do that is the walking and good diet that's low in sugar. You know, sugar is the enemy, not so much fat. And that you want to reduce stress. And the other thing that my generation did not get the memo for is sleep. Sleep is so important because that is when your system reboots. That is when the vacuum cleaners are flushing out things from your brain. And if you don't sleep well regularly, that system really glums up.
Okay. So sex differences are important, and it may be as you age they become less different, I suppose, is where we're going. So we're almost out of time. I think we've got a couple of minutes left. So I'm just interested to, you've both got Just a Minute, which is a very popular program here in the UK. I'm going to start with you, Mala, and then Janna. What's next? So what's the next big piece of work for you? What would you like to do next in a nutshell? So Mala, what are you going to, what's your, you're going to do a trial?
Oh yeah. Well, yeah, there's already a trial that's going to start that David Hafler and Jesse Cedarbaum are running with the currently FDA approved anti-TNF drug called infliximab. And it's an antibody to TNF. And they're going to do a trial. This is public information, so I'm not divulging anything that is proprietary. They're going to run a trial in our RBD and anosmics, and then we'll see what happens. We have a different drug that's slightly different than theirs, and ours is the one that I worked on in biotech. The difference is that the anti-TNF that we developed is only targeting the soluble form, not the membrane bound form that is necessary to fight infection. And so one of the things that happens when you take anti-TNF drugs right now is that you become immunosuppressed and you are more prone to TB and other infections. And so our approach is to selectively target the soluble inflammatory driving arm of TNF so that you leave the ability to fight infection alone. Ours is also more brain penetrant than the traditional antibodies, which cross a little bit, but not much. And so we're hoping that by covering both angles of selective soluble inhibition and brain penetrance, that will potentially have, you know, a good effect. Now, I'm really interested in their trial because their drug is mostly going to work on the periphery. So if they're able to slow the conversion from RBD to motor PD, they might be able to then have evidence that the periphery really matters for the brain.
Okay. I think that was just more than a minute, but I will accept it. Janna, you've got the last word on this. What's your next great experiment?
I think the next great experiment would probably be a lot of the animal models that we're using, especially in the inflammation field, tend to be on the younger side of things. I think what we really need to be doing is maybe hitting them in midlife and then letting them age all the way out and seeing what's happening along the entire trajectory. Like, what is the immune system doing that entire time? Where's the right time to intervene? Where's the right time or like, where are we seeing the major driving forces from the immune system? And particularly looking at those aged models. I know it takes a while to get them, but
Well, that's
we're,
great.
we're in an aging disease, so we need them.
We've got trials, and it's a question about timing and what we're going to target. So I want to thank both of you very much, Man and John. It's been a fascinating conversation about your lives and how we've ended up in this very interesting topic of inflammation, which is now moving into a new phase with trials and trying to better understand it to slow down the progression. Also for the insights into mentoring, which is something we haven't done much on the Research Spotlight, and it's so important to encourage the younger generation and to support all people in the field. I would like to thank Supernus Pharmaceuticals and Mitsubishi Tanabe Pharma America for their support in making this series possible. If they didn't support it, we wouldn't have a series, so we're enormously grateful. This is the last one of 2024 in terms of our Research Spotlight. We're starting again in February when Leonidas from Greece, who is actually the program chair for the next WPC, will be talking. He'll be talking about genetic synucleinopathies. He's done a lot of work on the alpha-synucleinopathies. In fact, the original mutation in alpha-synuclein comes from the Tansey family, which I think originates in Greece. So that will be a fascinating insight. So remember that you're free to join us on any of these. These are posted, so you can listen to them, anytime you like. Don't forget the 7th World Parkinson Congress, which is taking place on May the 24th to the 27th, 2026 in Phoenix, Arizona. So thank you all very much. Have a very good Christmas break, New Year, Thanksgiving. I'm a bit late for those in America, and see you in 2025.
Bye,
Goodbye.
everyone.